everyone, and welcome to today's podcast episode of Where Does Your Journey Stem From? Hosted by myself, Dr. Karina Minardi. Today, we are very fortunate to have Sophia Manukian as our scientific guest, who is a graduate student at Stanford University. Let's welcome to the stage, Sophia. Hey, Sophia, how's it going?
Hi, good. Thanks for having me today.
No, we're really excited to have you, so thank you. Sophia is a first-year PhD student in biochemistry at Stanford University, where she is currently rotating in various labs. Her previous research included her master's work on in vivo mRNA display, a high-throughput technique developed in the Tavazoie Lab at Columbia University to study protein interactions. When not playing around with proteins, Sophia enjoys participating in STEM outreach and mentorship, which she has been involved in since her undergraduate days at UMass Lowell. In her spare time, you can find Sophia climbing at the local rock gym or enjoying a mug of herbal tea. Delicious. So again, Sophia, we're so excited to have you. And as we always start off a podcast episode, tell us a little bit about you as a person, your background, and so on and so forth.
Yeah. So I grew up in Massachusetts, and I kind of started my scientific journey, I would say, in high school when I was fortunate enough to have a teacher who was also a PhD student at UMass Lowell at the same time, which is kind of amazing. And she was able to introduce me to the science that she was doing as part of her PhD. And later, when I attended UMass Lowell, I actually joined the lab and worked on a project. And this, I think, was my first adventure into the world of scientific research. And near the end of my time at UMass Lowell, I started to think about what to do next. And that was a difficult decision for me that I thought about for a long time. I didn't know whether to do a PhD, whether to do a master's, to go straight into industry. I had a lot of people kind of suggesting that I go straight into a PhD, but at that time I didn't feel ready. But I was really lucky to receive the National Science Foundation Fellowship for grad students, which allowed me to actually go to Columbia University and do a master's degree. And this was a really great opportunity for me because it gave me the chance to explore science and really broaden my knowledge. While I was there, I joined a systems biology lab, and when I got to Columbia, I didn't even know what systems biology was. So it was a really great opportunity for me to learn more about science and also learn more about myself. And this really invigorated my passion to continue pursuing scientific research and apply to PhD programs. And so that kind of brings me to where I am today, which is in my first year of my PhD at Stanford in biochemistry.
That's a really exciting journey. And so there's a couple of things that I want to deep dive with you on. So that teacher in high school, what was that about him or her that lent itself to really providing you sort of an introduction to research and science, STEM?
Yeah, that's a great question. I think mentors are really important and kind of what brings people into science and can encourage people to go down different roads. And my high school teacher, she was just really a great and enthusiastic person. And everything that she did, she kind of tried to tailor specifically towards students and like our level of understanding, and tried to like work with everyone to make sure that she was taking the information, the complex information she was trying to share with us, and like breaking it down so we could understand. And she was also really encouraging during the whole process, because she always kind of acknowledged that like this is a complex thing and you're just starting out, and so it's okay if you don't understand or if you make mistakes. And I think that's really important when you're first starting out doing anything.
Well, everyone makes mistakes, and I think it's kind of funny because I always like to say that, at least in my research background, that it wasn't necessarily the mistakes that I made, but it was when I had experimental results which were either the complete opposite or counterintuitive to what I was hypothesizing, and that was really how I learned. So it's really beneficial, I think, for me. And then also the piece around your mentor of saying, you know, you're taking something that's super complex and then actually, you know, providing sort of a baseline assessment of how to convey it to students, because this is the first time that they're just hearing it, and you can't just expect them to memorize something. So I think that's a wonderful methodology. So I want to delve in, though, fast forward then to your master's and your introduction to systems biology, because it sounds like you didn't quite know what systems biology was before you went to Columbia, and that is a great leap of faith, for lack of a better word.
Yeah, I think that it was to a certain degree, but when the work that I did as an undergrad, I was working in a protein biochemistry lab. We were studying the muscle protein titin, and kind of most of the work being done in the lab was on this specific protein. And we were looking at one small, and this protein is gigantic. It's the largest known protein in the human body. And so I was looking at a very small part of this giant protein. And then kind of fast forward to when I got to Columbia and I learned about this lab. And during my work there, I was working with a library of proteins. So I had over 10,000 proteins that I was working with at the same time. And so that was really exciting to me, to go from like this very specific niche system to like, let's think a lot more broadly and in a really different way. And I think part of the reason I took that leap of faith into something that was new for me was because it was a master's program. I knew I wasn't going to be there for very long, and I knew that I could really like jump out and try something new and explore. So I think that was really great that I had that opportunity and chance to try.
No, that's a great point, I think. And then we're obviously going to talk a little bit about you're currently making your decision around PhD. So table that for right now. But I think that that's a great sort of juxtaposition to where you are right now, right? So talk to me a little bit about, let's actually just deep dive directly into your undergraduate research. You know, what were your hypotheses? What did you prove to be true? What were some of the instrumentation you used?
Yeah, so the main project that I worked on during my time as an undergraduate was looking at this intrinsically disordered motif in the muscle protein titin. And so titin has a lot of different domains, but there's one section called there's one section that has this intrinsically disordered region that has all of these poly-Es, so a lot of glutamic acid residues. And we were trying to learn more about the sequence of this region and how it reacts to pH, because upon muscle activation, there's a decrease in pH. And so we were trying to learn how differences in sequence can lead to a different conformation of this muscle protein that can act as kind of the molecular ruler of the sarcomere. And we were doing that by taking different peptides that had the poly-E motif and changing the uncharged residues to see what impact they have on the conformation.
And can you explain for the listeners what an intrinsically distorted motif or intrinsically distorted protein is? Some of my colleagues in grad school actually were looking specifically at IDPs, and so I know what it is, but it's a really cool, interesting subsection of biochem.
Yeah, I agree, actually. So an intrinsically disordered protein is just, or an intrinsically disordered region is just a part of a protein or entire protein that doesn't have one specific three-dimensional shape. And I found this really interesting because kind of in basic biology, biochemistry classes, there's kind of this dogma or thinking, one protein, one function, or the function of the protein is tied to its three-dimensional conformation. And then when you start to think about, oh, this protein doesn't have one set conformation, it can be in a bunch of different ones. It can change its conformation based on the conditions it's in or what it interacts with. And so this was really interesting to me because it kind of defied the kind of really basic introductory thing that they teach you in biology class.
Yeah, I think that when I was in biochem back in the day, it was structure-function relationship. And, when I went to grad school and I was introduced to IDPs, it was like, oh, that just went out the window.
Right.
So it's a fascinating topic, personally. So then fast forward then to your days at Columbia with respect to systems biology. You have a library of proteins, were some of them IDPs? What were you actually doing? Were you creating a catalog? Let's deep dive on that now.
Yeah, so at Columbia, I worked on in vivo mRNA display, which was something that was already developed in the lab in a yeast system. So basically, how this method works is it's a method to try to understand protein interactions and to try to understand them at a massive scale. So instead of just testing one protein against one other protein, what if we could test one protein against a whole library of 10,000 proteins at the same time? And we already have the capability to do this with some methods like mass spec, but that comes with its own challenges. So the idea with this technique was to create a plasmid essentially that allows a protein to be tagged with its own mRNA. And then you can sequence that mRNA in order to identify the protein as opposed to using mass spec. And so this kind of leverages the improvements that we've seen with next generation sequencing technology to also be able to look at proteins, which are notoriously kind of, at this moment, more difficult to study in a large scale. And my work was focusing on applying this, making a library of human proteins, because the method had already been developed in yeast with a library of yeast proteins, but in order to probe questions about human biology, you need a library of human proteins. So that's what I was helping with.
So what was the methods by which you used? I'm a little curious about that, because I would anticipate there to be computational, or at least that to have some sort of level of activity, in addition to being, lab bench, traditional work, maybe even PCR, I don't know.
Yes, so a variety of things went into this project. So first, there was a lot of lab work to kind of we had a library of clones that had open reading frames or ORFs on them. So essentially, each clone would have a protein, and we had to take all of those proteins from the plasmid that they were in and put them into the mRNA display vector. And so that was a lot of cloning and expression work. And afterwards, what you can do once you have this library is you can do an IP, or immunoprecipitation, in order to pull down a specific set of proteins and then isolate the RNA and then sequence your sample. And afterwards, you look at both a control where and a sample where you've pulled down something specifically, and you see what is the amount of reads you have in one sample versus the other. So if you have something that is has a much higher number of reads in your sample where you've pulled something down, then you can say that, that is being pulled down in this experiment. And so I was more involved with the bench rig aspect of this project, but after you do the pull down experiment, there is a fair amount of computational work that you need to do to analyze sequencing data. I wasn't fully involved in that aspect of the project just because of the time constraints, but I kind of got to learn about what the workflow was for doing that.
Well, I think, though, that it really is an example of it takes a village to complete some of these experimentations, because it is so, it is so dependent on what your subject matter expertise is, as well as, to tell a full and complete story of what you are actually looking at. You are going to need your pure basic sciences, in addition to, the new technology by which you're building, but then also the theoretical of the computational, right? And not everyone can be, I think, an expert in everything to really get that experiment going. So I think that's a great example of not only collaboration, teamwork, but also that it honestly, it does take a village.
Yeah, I think it can be really helpful to have people with different expertises, and this is why collaborations in science sometimes can be really fruitful, because you can take the time to try to learn all of these things on your own, or you can work with someone who is an expert in that thing, and that can, depending on your project, be very helpful and like a better use of everyone's time because they know exactly what they're doing.
Well, and I think this is a great segue too, because I would anticipate that this would be part of your deciding factors on when you're thinking about a PhD lab. We had a couple of previous guests on in which they were in labs that were very niche science, and so they were very focused on specific, opportunities. But then we also had, frankly, some guests who said, we have biochemists, we have biologists, we have chemists, we have p-chemists in our lab, and that was really a deciding point for the guests to make their determination in a lab. So let's segue then into your current state. So you're doing rotations, you're in your first year. Tell us a little bit about kind of what is in the daily life of Sophia.
Yeah, so I am currently in rotations and taking some classes as part of my program. I recently finished my first rotation and started my second rotation. And my first rotation was focused on, was a cell signaling lab where I was thinking about a protein called NPC1 as part of my project there. And now I'm working in a lab that studies cell death mechanisms. So it's been really interesting to be able to think about and sample a lot of different kinds of science. And it's also been interesting because as I've been in classes and also trying to get something done in my rotation projects, there's also kind of this constant in the background need for like learning what's going on at Stanford and like what other labs am I going to rotate in? Like what kinds of things am I interested in science? Like what are things that I need or am looking for in a PhD lab? Is this important to me? Is that factor important to me? So there's, in addition to kind of like the daily things, daily tasks going on, there's always this kind of background of like, what do I want to do? Where do I want my PhD to be?
So talk to me a little bit about your first rotation versus your second, because I could imagine that cell signaling is quite different from cell death. Maybe it's the same, I guess. I guess there are some cell signals for apoptosis, now that I think about it. God, I'm reaching back into my memory for that from my biochem days. So there's the science, right, which is the research topic, and then there's the environment, and then there's the what do you do with this research, long term, post PhD.
Yeah, so I think my first two rotations have already given me the opportunity to like compare and contrast some things. I think the environments have been slightly different. One of my first rotation was a very kind of postdoc and scientist heavy lab, whereas my second rotation has mostly graduate students. And I think that lends itself to a slightly different environment. I don't think that one is necessarily bad or better than the other, but they are mildly just different. And it's also been interesting to see my first rotation. I think the point of view from the lab was a little different than the second one. In my first rotation, they were very focused on kind of protein biochemistry and like signals. There were a lot of projects in the lab that were like, there is this protein, we know it's involved in some way in this process, but we really have no idea what this protein really does or how it is doing that thing. And so a lot of projects were kind of like a black box of like, what is going on with this protein? Whereas I think the second lab also thinks about proteins, and cell processes and how the two, how proteins are important for cell processes. But I think they think about how do other molecules interact with proteins and like lead to the cell process. So they're thinking about cell death, and some of the work that the lab has done or has thought about is different molecules that inhibit or lead to cell death. And so it's kind of a little bit of a different thinking style, like how can we perturb this system and lead to different outcomes.
That's fascinating. And so how long is a rotation? And then are you thinking about, and then how many more, I guess, do you have to do?
So I think different programs handle rotations differently. In my specific program at Stanford, I'm really lucky that there's a lot of flexibility. So I think the kind of standard thing is to do 3 rotations and 1 rotation per quarter, and each quarter is about 10 weeks or so. And that's something that I think is kind of the standard, but my department is nice in that it's very flexible, and they said you can do shorter rotations if you would like. And so that's kind of what I've chosen to do, because I think that the main goal of a rotation for me is to figure out if the place would be a good place for my PhD. And I think I can learn what I need to learn about that in 6 to 7 weeks. And I don't need the full 10 in order to do that. Although some people like to do longer rotations because they really get interested in something, or they're like, oh, if I worked on this a little longer, I could get on a paper. But for me personally, I think the goal is to learn a lot and find a home for my PhD.
Yeah, and most of the time you can very quickly identify that, I think, in 6 weeks, to your point. You can tell by the environment. You can interview all of the different people that you're working with. And then you can also take a historical perspective on where the research was. And, I think a huge piece there is, how long does it take traditional students to get through? So on and so forth. Those sort of things that you need to get through. So what do you, I guess, as you start choosing your PhD, how are you thinking about post-PhD in a career?
I think I have always been kind of interested in industry and the biotech and pharma industry, especially growing up in the greater Boston area. I've kind of been exposed to that environment for a while. So I think that's my eventual goal. But a piece of advice that I got from one of the faculty here, and I think I really I think I really value this piece of advice, was right now just focus on having a good PhD and learning something and doing well in your project. And then later on, you can really think about like, what do I want after, and make that happen. So I think my focus right now is just having a good PhD and learning all the things that I can possibly learn.
Oh, I think that's spot on. It is quite helpful, however, to think through, how does your PhD, whether it's your mentor, your research area, your lab in general, really align for your long-term aspirations and goals. So I know that's crucial, but I think also, though, a PhD is an investment. You are investing 5+ years of your life into something, and so you better like it.
Right.
And or you better enjoy, for the most part, because you won't enjoy everything. That's impossible, frankly, impossible. But you better enjoy most of it, and the ride. So I appreciate those words of wisdom. So I guess, reflecting on your story, what words of wisdom would you impart onto future STEM students?
Yeah, I think a few different things. One thing, kind of going back to the point that I just mentioned, which was I think that there certainly is a lot of value of looking forward and thinking about where you want to end up and how to get there. And I think that's really valuable and something we should all do. And it's really useful to think about where you're going and how to get there. But I also found that I, at certain times, have been overwhelmed by the amount of things to like think about, and like, how are the decisions that I'm making now going to affect my future career and all of these things. And a piece of advice that I got that was very valuable to me as I was making some important decisions, like whether to do a PhD or a master's and all of these things, was, what do you want to do now? Like, what makes you passionate now? And when I was thinking about doing a PhD and applying for PhD programs, what made me passionate then was learning about science and continuing my journey to become like a rigorous scientist. And that kind of thinking really helped take away all of the other factors that were kind of bombarding my brain about, like, how is this going to be for my career progression? What if I don't like it? What if I'm not good at it? All of these other factors, and like simplifying the question really helped me in that moment. And then I think one other thing that I would say to younger scientists is to rely on the network of people around you in science. I think it's really important to ask people for help as you're going along. And I think this is something I could have done a lot more as I've been going through kind of this process of kind of trying to become a scientist, is go out and ask people around you who have gone through what you're going through for advice. And that's kind of been one of the really nice things I've noticed since coming to Stanford, is almost every one of the grad students that I've approached to ask about labs, to ask about their experiences, to ask for advice have been really wonderful, and taken the time to sit with me, to talk with me, to give their perspective. And I think that sort of thing is really invaluable because they kind of have the hindsight of having gone through it already. And so many people are nice and helpful. So really, don't be scared to go out and try to ask people for help and guidance.
No, I really like that. And I like it because the worst case scenario is that they will say no. And if that is the case, then you don't want guidance from them. And so, it's a win-win, frankly. You just have to be brave enough in order to ask it. So I really like that answer. And there's a lot of different perspectives, and I think getting more information than less information is always very helpful. And yes, it may bombard your brain, as you said. It also, though, helps you make an informed decision, which is what you need to do in life. And you will continue to do this in life throughout, decades, right?
Yeah.
Ideally. Well, with that, Sophia, I thank you for your time and telling us a little bit, not only about your story, but also yourself. And so I appreciate you. And again, to all of our listeners, thank you for tuning in today. And always remember to ask yourself, where does your journey stem from? Bye, everyone.
Thank you.